Okay, everybody, welcome to Lecture 17. Today we're going to talk about lipid biosynthesis. This is a pretty dense slide deck, but I don't think always necessary, so I'll try and pick up the ones that are important, and we'll go from there. So from this, we're going to be talking about fatty acid synthesis, so this portion here, and lipids and all that stuff, so let's go ahead and talk about that. There's many different kinds of lipids. We've got all sorts of things. We've got triacylglycerols, we got cholesterol, phospholipids, free fatty acids, and they're characterized. Lipids generally are characterized by low solubility in water, and they have this like amphipathic nature where they're I believe like when we talk about amphipathic, we're generally referring to my brain. Where it's both hydrophilic and hydrophobic, but in the sense that like one side is hydrophilic and the other is hydrophobic. So like with these kinds of tails, right? The big deal here is that this part is hydrophilic, but the rest of it, is hydrophobic. So that's what I mean by amphipathic. And there's a strong tendency for them to aggregate together in aqueous environments because, in this case, like dissolves like kind of thing, right? The things that are hydrophobic will interact with other things that are hydrophobic, and they'll kind of repel things that are hydrophilic, if you will. So the general structure of a free fatty acid is a polar head group followed by an aliphatic tail. An aliphatic tail is just a chain of carbons, like a massive chain of carbons. That's it. There's some numbers here if you're interested, but I wouldn't be too worried about them, really. These chains can be really long and they can be really short. Doesn't really matter. It's a carboxyl group, hydrophilic, long hydrocarbon, hydrophobic. That's what makes up a free fatty acid. It's also saturated versus unsaturated. So if you remember from OChem, like saturation number, stuff like that, if it's saturated, it has all the possible hydrogens that it could have. If it's unsaturated, it's missing hydrogen. See, like here, there should be an H here and an H here. But because there's a double bond here, we're missing that. And what that actually does, it actually ends up bending the molecule. Remember, because double bonds are cis or trans. So this image is actually a bit misleading because it depicts it as straight. But actually, instead of going straight like this, it should really like either this section of this should actually be angled like up or down or something like that. That would be more accurate. There's also polyunsaturated fats, so you could have, this only has one double bond, but you could have two double bonds, you could have three, you could have four, it doesn't really matter. You can have as many as you really want. So there's monounsaturated, so only one double bond, and polyunsaturated. And these fatty acids are weak acids, and that should make sense because this is the head group, which is a carboxylic acid. And, they're usually going to be, they have a pKa of 4.5, which is, acidic compared to the, physiological pH of around 7, so they're going to be negatively charged at physiological pH. I talked about this already. They can be in cis or trans conformations. So this is just saturated, this is cis, and this is trans. So cis introduces this 30-degree kink in the chain, so this is like a 30-degree angle right here, if you will. And the trans is this image here. So I mean, these are just cool things to keep in mind, and you want to definitely remember them. Fatty acid hydrogenation. So trans fatty acids, this is a cool slide. I don't think it's that important. You find these trans fatty acids in different things, but cool. For fatty acid nomenclature, the way that we describe it is we say, okay, we use this delta nomenclature. And so what we do is we say, there's two. There's delta and then there's the omega nomenclature. So the delta nomenclature is where we, how many carbons are in the chain. So if we look at this one here, there's going to be 18 carbons, so there's 18. How many double bonds are there? There's 1 and 2. And the delta just means the double bonds. So where are the double bonds? So we put delta 9, 12. So it's on the carbon 9 and carbon 12. So that's what it is. So 18, 2 parentheses delta 9, 12 means we have a fatty acid that is 18 carbons long with 2 double bonds. And those 2 double bonds are on 9 and 12. The other version of nomenclature we have is the omega nomenclature. And this one is not as useful, I would say, as the delta nomenclature, because it doesn't tell you where all the double bonds are. But it's helpful for figuring out where the last one is. So we can see here with omega, we still do the same thing, 18 and 2, but instead of counting a delta 9, 12, instead what we do is we look at the omega. And omega refers to how far away from the last carbon is the first double bond. So this guy right here, the nearest double bond is 1, 2, 3, 4, 5, 6 carbons away from the last carbon. So it's going to be an omega 6 fatty acid. So that's why we call with this weird W and 6, right? Okay, so for free fatty acids and all this stuff, this should hopefully make sense. The physical properties, like the melting point and stuff, are determined by the length and the degree of unsaturation. And the reason for that is because it basically affects the density, right? So different degrees of packing, and that influences solubility and melting point. And so if we look at like this graph here, you can see that, as we, these are all saturated fatty acids, right? As you increase the number of carbons, the melting point goes up, which should make a lot of sense because there's going to be more intermolecular interactions, not reactions, interactions between the different hydrocarbon tails. And so if you have more interactions, they're gonna have a stronger force to attract to each other, and therefore they're just going to combine and have a much higher melting point. On the other hand, though, if you have if you start adding double bonds and stuff, so like we can look at this graph instead. Now we've got a saturated, trans, and then cis. And so what you notice here is that the saturated one has a much higher melting point compared to the trans and the cis. And this should make sense because remember, with saturated fatty acids, they're very tight interactions. With trans ones, right? Remember, trans. See, these trans guys, they look very similar to the saturated ones, except they still have a double bond, which means that they're not as fluid, right? They're not as malleable and able to compact quite as well as these saturated ones that are just able to move around however they want. So they're a little bit more rigid, and so therefore their interactions aren't as strong, and because of that they have a drop in the melting temperature compared to saturated fatty acids. And then for cis ones, it should make a lot of sense why these are much lower. These things lead to massive gaps when you combine them together. It's like, see like how there's no interactions in this space at all, no interactions in this space, no interactions in this space, that kind of stuff, right, where there's no interactions, and because of that lack of interactions there, it drops the melting point significantly. So now let's talk about how we synthesize fatty acids. So this is an example of palmitate, which is a 16-0 fatty acid. And the way that we do this is actually through acetyl-CoA. And so acetyl-CoA, remember, comes from pyruvate dehydrogenase. It goes in the citric acid cycle normally. But we actually end up using this to kind of kickstart our formation of fatty acids. So, yeah. So here's fatty acid synthesis as an overview. Basically what's going to happen is we're going to take some amount of acetyl-CoAs, we're going to add some CO2s, some ATPs, some NADPHs, and we're going to make this really long chain. And if we want to break this chain down, we can do beta oxidation and break it down. But what we have to remember, right, is acetyl-CoA is in the mitochondria. So we have to get the acetyl-CoA actually out of the mitochondria because fatty acids are actually stored in the cytosol, not the mitochondria. So a citric acid cycle occurring in the mitochondria means that we have to, take out those acetyl-CoAs and put them into the cytosol so that we can actually store them as fat. So the three key steps are that we have to first shuttle the acetyl-CoA into the cytosol. We do this through the citrate shuttle. Jeez. We then have to form a activated version of acetyl-CoA, which is malonyl-CoA, and we have to do that because otherwise we're not going to be able to like combine it into a polymer, right? Because acetyl-CoA, it's not extremely stable, but it's a pretty relatively stable molecule. So it's acetyl-CoA carboxylase, right? So we have to use acetyl-CoA carboxylase to activate it so that we can actually use it as a kind of a... so we can use it to create fats. And then we have to build the actual palmitate, or the 16-carbon fatty acid, or 18-carbon, whatever you want to build, right? So we would do that using fatty acid synthase. So here is the citrate shuttle, and honestly, I could yap at you about this for a while. I think the easiest thing to do is to just go through a round of it. So here we're in the mitochondrial matrix, and you know, citrate is made through the combination of oxaloacetate and acetyl-CoA. So we have this here. So oxaloacetate and acetyl-CoA. These maybe our body has some acetyl-CoA in it, and they combine to make citrate, right? And citrate, what ends up happening is citrate will actually go through this citrate transporter out into the cytosol, and then we have an enzyme called citrate lyase in the cytosol, and this thing cleaves citrate into oxaloacetate and acetyl-CoA. This oxaloacetate is then reduced to malate, and then this malate can undergo, I believe there's two different options. The malate can either go through the malate alpha-ketone glutarate transporter, or and then go through malate dehydrogenase to turn to oxaloacetate again, or it can go through the malic enzyme, which actually is a decarboxylation enzyme that changes this into pyruvate, and then this goes through a pyruvate transporter, and then this goes through pyruvate carboxylase to make oxaloacetate. So there's a couple ways you can do this. This is like a slide I feel like you could probably put on your note card. There's a lot of steps here. I don't think you have to know every single step here. I think it's a lot, but hopefully that kind of makes sense. Essentially, all we're doing is we're using oxaloacetate as like an agent to carry acetyl-CoA out from the mitochondrial matrix into the cytosol, so that we can then use the acetyl-CoA for fatty acid synthesis. And then on the way back, there's two options, right? After we've cleaved this using citrate lyase, and we're left with oxaloacetate, we can either after we have this oxaloacetate, we can reduce it to malate, and then from here we have two options. We either cleave it and release the CO2 and turn it into pyruvate, or we just shuttle it straight through the malate alpha-ketone glutarate transporter and oxidize it to oxaloacetate. Right. So we can do either one of those. So malate goes straight to here, you oxidize oxaloacetate, or you can turn to pyruvate, carboxylate the pyruvate, and then go to oxaloacetate. So a couple different options here. And the reason why this is kind of helpful is because of where is it? This. We tend to do the pyruvate pathway because it generates NADPH, which actually is used in the process of making fatty acids. So we tend to use the pyruvate pathway when we're doing these kinds of things because of the fact that we need this NADPH to actually make fatty acids in the first place. But it doesn't mean that it can't do this pathway. If you don't need NADPH, then you can just do that pathway, right? So why is citrate available for export? So you're probably thinking, okay, well, if citrate is in the citric acid cycle and the mitochondria, which wouldn't it just go through the citric acid cycle enzymes? And to answer that, you have to remember that, a lot of these enzymes are inhibited. So like isocitrate dehydrogenase, alpha-ketone glutarate dehydrogenase, these are enzymes that are inhibited. So with that in mind, gosh, I'm like yawning already. What's going on? With that in mind, basically what's going to happen is, if you have high levels of energy, like high levels of ATP and ADH, these will inhibit a lot of these enzymes, and so therefore this citrate kind of just like gets stuck. And you can't really use the citrate, and so when you can't really use the citrate, sorry, I'm just like turn my volume down a little bit because I'm really, I think it's peaking a little bit. Okay, hopefully that's better. But okay, so the citrate, you know, doesn't have to be in the citric acid cycle if you already have enough energy. So that's why it's able to actually get shuttled out. And now we get to talk about acetyl-CoA carboxylase. This is a really cool enzyme, yeah. I'm going to skip this slide, actually, and just talk about this slide. So, hold on a minute. Why is it called step? Oh, yeah. I'll talk about this slide. So this slide is the one that I really care about. So, basically, let's see here. This is bicarb indeed. Yes, okay, cool. So, basically this occurs in like 4 steps. So, we first have to make the acetyl-CoA into its activated intermediate, which is malonyl-CoA. And the way that we do this is through acetyl-CoA carboxylase, like I said. So this enzyme uses an enzyme called biotin, and biotin, I've talked about this before in 440, is just general enzyme responsible for carboxylations the majority of the time. TPP, on the other hand, is usually responsible for decarboxylations, but also is important in transamination reactions. So you see biotin, think carboxylation. So if we look at this, basically what's happening in the enzyme in step 1 is that we take this bicarbonate, because we don't just have gaseous CO2 running around that we can just extract from the air and just like use to carboxylate. We use bicarb and we use ATP to make this activated bicarbonate. And then what happens is that this biotin molecule actually does this kind of attack where it forms a bond with this bicarbonate, and you get this intermediate shown here in 2. And this happens in the biotin carboxylase domain. And then after this, what happens is that this arm here, this biotin carrier like protein arm, swings around to 3 here. And you can see here how the arrow swings around this way to the trans-carboxylase domain. And what happens here is once this biotin hand thing has flopped over to the other side with its CO2 attached, the acetyl-CoA is able to dock into this site, and then that results in a kind of reaction where this acetyl-CoA attacks this carbon and kicks off this piece of the biotin, and so you end up with malonyl-CoA. So now this is an activated malonyl-CoA, and this should make sense why this is okay and favorable to do, because we've used ATP as a way of driving it. So we've hydrolyzed ATP to activate this malonyl-CoA, and that is why this reaction moves forward. So regulation of acetyl-CoA carboxylase, this is the main one that gets regulated. So fatty acid synthase actually, if I'm not mistaken, does not really get regulated. I think it just doesn't get regulated, period. But we can go back, we can go and take a look at that in a second. But it's the main one. So basically, these inhibitions and activations should make a lot of sense. So it's activated by citrate. Makes sense, right? Citrate is a substrate. It's inhibited by long-chain fatty acids. That makes sense. The final byproduct of this reaction, or the final product of this reaction is long-chain fatty acids. So makes sense that it would inhibit it. And then in terms of hormones, we have to think about what each of them does. So insulin promotes storage, right? And glucagon promotes the need for sugar. So insulin, right? This process is for storing energy. So insulin activates it. So insulin, which leads to dephosphorylation generally, right? So dephosphorylation, that means, of ACC activates it. On the other hand, epinephrine and glucagon, they are signals that you need to create energy and use the energy, and so they would actually inhibit the carboxylase, because you don't want to be making energy stores when you need to be using it in the first place. So that means that phosphorylation of ACC leads to inhibition, right? Because glucagon leads to phosphorylation. And these phosphorylations, they generally occur at things like serine, threonine, and tyrosine. That should make sense because, I mean, those are amino acids that have OHs on them, right? And, you know, you can phosphorylate those pretty easily because they have the OH. This is just a cool, kind of like some data on acetyl-CoA carboxylase. I don't think it's that important, but I think the main thing is you should be able to like understand like how this works. Like if you have citrate and it's phosphorylated, there's some level of activity. It's not great, but then when it's dephosphorylated, it's like crazy levels of activity. And then for ACC inhibition, if you add a bunch of long-chain fatty acids, it inhibits it. Cool. Makes sense? And yeah, now we get to talk about fatty acid synthase. So fatty acid synthase is really interesting. It's a really long process. And so essentially what's happening is it requires 6 different enzymes activities present within fatty acid synthase. So fatty acid synthase is like a 6 enzyme complex, if you will. And it's a single polypeptide with multiple catalytic domains. So it's like one massive chain, but like it's like 6 different enzymes. It's kind of weird to kind of explain, but think of a really long protein that has multiple catalytic sites and acts as different enzymes all at once. So we've got a couple. So we have the transacylase, the synthase, the reductase, the dehydrase, and another reductase. So these names should be self-explanatory, right? Transacylase is a transfer reaction, synthase is some sort of synthesis, reductase, some sort of redox reaction, dehydrase is removal of water, and reductase, again, is some sort of reduction reaction or redox reaction. So let's go ahead and get into it. I was hoping that they would do each one individually. Yes, okay. So, priming. So, we're first at the malonyl acetyl-CoA transacylase. So what we're doing, right, and they've actually labeled the carbons here to make it easier to track. Basically, we have this ACP, and I believe, did they name ACP for it? I can't remember what it stands for anymore. It's been a while. I think it's acyl carrier protein is ACP. So, basically what happens is this acyl carrier protein is going to have this malonyl-CoA attached to it. And this is done through kicking off that CO2, if you remember. Well, not kicking off CO2. Sorry, that's later. So first we need to attach a normal acetyl-CoA to this. Basically, it's a transesterification. No CO2 involved, no malonyl-CoA. This is just a normal acetyl-CoA. So a normal acetyl-CoA is attached to this ACP. And then what happens is this ACP transfers this acetyl-CoA to the KS site, so the ACP synthase. And so from here, what happens is, once we've transferred to this KS, this synthase site, which basically acts like a dock, what we do is we now start doing things with malonyl-CoA. So we take a malonyl-CoA, we attach the ACP to it and kick off this S-CoA group. So now we have this. And then this swings over, so that happens in the MAT site, and it swings over. And then we end up adding this to our chain. So, if you see here, basically, these electrons go back to the carbon dioxide, or the CO2, and this bond breaks and attacks the carbonyl here. And so when you do that, this then comes back down, right? So when you attack this, these electrons go up, and then eventually they'll come back down, and this bond here will break. And that is exactly what happened, right? See how the KS bond broke? And now we have this guy. And now,
since we're here, what we can do is we can use an NADPH molecule to reduce this OH group, or this carbonyl group, to an OH. Then we use a dehydrase, and a dehydrase is literally what it sounds like. It removes the OH and an H, right? So we're gonna remove these OHs, and also some. We're gonna remove an OH from here and the H from right here. So we're gonna release water, as you can see here. So we form a double bond. And then we use a reductase, which uses another NADPH. So we have two different NADPHs being used here. So then we have just this fatty acid chain. And then it goes back here. And then this whole chain is added to KS. And this process is then repeated again. So then you take another malonyl-CoA, you attach it to ACP. You then do this thing again where you knock the electrons back. These guys come here and attack here. And you can see here, like, eventually if you just over and over again, you get this really long chain, right? And then that's basically the main deal there. And so how does the question is, how does the synthase know when to stop? Why doesn't it just make like an 8 million long carbon thing? And the termination is based on the specificity of KS. Versus the termination domain. So let's see here. I'm trying to see if it tells you exactly. Basically, there's like a length. Yes. So basically, it can only accept saturated acyl intermediates for elongation. So basically, there can't be any double bonds in it, otherwise it doesn't fit into the enzyme properly. And so that's how it's able to differentiate. So if it has double bonds in it, it doesn't actually fit into the enzyme, and so therefore that's how it kind of figures out when it has to stop. So trying to see if there's like a binary acyl chain is not energetically favorable until the acyl chain reaches 16 carbons. Yeah, I was trying to see if they kind of explained it, but basically, I think the main thing you should know is what I said before. Like, it'll only accept, so KS can only ever take saturated acyl groups, and then the 16-0 acyl chain is a poor substrate for KS domains. And so basically what's happening is once you form this like 16 carbon long fatty acid, it doesn't like go back into the enzyme because it's kind of too large. And also, like I said before, double bonds don't really fit. So it's a size thing as well as a shape thing. So if you have an unsaturated fatty acid, it doesn't really fit very well because of the shape of it. And then for 16 carbons, that's just the maximum length pretty much. It's just, that's how long it is. And you can add more to it later, but that's pretty much the max length that's the synthase is able to handle. So just because of size constraints from what I understand. So here's a nice little slide that I would use to kind of memorize and get used to fatty acid synthase, but yeah, I'm not going to go over this whole thing again since you guys have already talked about it. So just remember, like, that in order to do this thing where you form the big chain like this, you have to do the cycle like quite a few times. So every cycle you do adds 2 carbons, right? So basically you're adding, so we started with these 2 and then we added these 2, right? So like if we look at, let's start here. So we started with, where's the starting point? Started with these 2 initially and we added these 2. So basically, if you take the number of carbons and divide it by 2, then you have the you basically have the number of cycles that you had. Or you should be subtracting the first 2, because the first 2 obviously came from the initial acetyl-CoA, and then everything else after that came from a cycle of the actual synthase. So if I have a 16 carbon long fatty acid, 2 of them came from the initial acetyl-CoA, and the other 14 came from the transacylation and the whole circular pattern. So you have to use 7. You have to use 7 times 7 cycles, which is 2 NADPHs per. So it would be 14 NADPHs, and you'd also be releasing like 7 waters-ish. And I think we talked about that up here. But if you go down, there you go. Cycle repeats 6 more times. I believe I'm trying to see if what they did here exactly. But yeah, 6 more times, 7 total cycles. See, if you want to make a 16 carbon long palmitoyl-CoA, or palmitate, not CoA. So palmitate can be modified, like I said before, to generate more fatty acid types. So the elongation increases the fatty acid chain by 2 carbons, and the elongation occurs from the carboxyl end. So like the COO end, and it uses malonyl-CoA but not fatty acid synthase or the acyl carrier protein, so it's a bit of a different mechanism that you don't really have to worry about for this class. You can also talk about desaturation, so generating cis double bonds using desaturases. So yeah, that's something interesting to keep in mind. Like you can add there's enzymes that add double bonds, so if you look at this, elongation occurs from carboxyl into the fatty acid group. So we have, see here, we can, this is just a point that they made. But you can see here that you're adding another desaturation here, right? So you're adding one there. You can actually move, like you can basically, because you add carbons from this end here, look at, if you see it's initially, right? There's an enzyme that always adds a what's it called? Adds a double bond to position 12. It looks like in this case. So or sorry, not 12. It looks like in this case they're looking at position 6. But basically, there are certain enzymes that are able to desaturate at certain positions on these kinds of chains, and some of these desaturators are only present in either humans or plants. Plants usually have more desaturases because they have other needs for fatty acids. But if we look at this, you can see that okay, so we desaturate on carbon 6, and then when we add a new like acetyl-CoA, we're adding it here, and so what happens is these carbons shift up by 2, and that's why these are on 8 and these are on 11 and these are on 14 now. So, and then if you want to desaturate at 5, you can do the same thing, right? You desaturate at 5, and if you add 2 carbons again, right? If you add another 2 carbons, these will shift up to 7, this will go up to 10, this will go to 13, and this will go to 16. That's the kind of thing that you should keep in mind. And this is something I would memorize. Humans have 4, 5, 6, and 9. Plants have additional ones, which are 12 and 15. And the other cool thing about these pathways is the arachidonic acid pathway, actually. So linoleate, which is 18,2,9,12, omega-6, is the precursor for arachidonate, which is the precursor for eicosanoids. And these are really important because they actually do a lot of cell signaling. Arachidonic acid, or they're not called arachnoids. I think it's arachidons. There's like a name for them, but they're like a class of molecules class of signaling molecules that are like very important for, and very, very importantly derived from these linoleate. I believe like phospholinositol phosphate 3, so PIP3, PIP2. I think those are arachidonic acid derivatives, but can't remember off the top of my head. It's not actually listed here, so don't quote me on that, but yeah. And here we're just talking about, the different, here's different names for these. I would say palmitic acid for sure is one you should memorize. I don't really think it's important to memorize the names for all of these. It might be worth putting on your note card if you're really worried about it, but I don't think it's that important personally. But other than that, actually, we're actually done with the video. So, I hope the video was helpful. I was on a bit of a longer one, but tried to make it succinct and easier to follow. I know it went a bit fast, but I'm just trying to make sure I get these videos out. So, yeah, I hope this was helpful, and yeah, happy studying.